We'll begin this session with a presentation from Stefan Huhn, Vice President, Clinical Research and Chief Medical Officer at StemCells Inc. Dr. Huhn has been responsible for the oversight of the neural stem cell clinical research and program for the last 7 years, which includes all of the past and ongoing trials. These studies have involved the first human trial for disease affecting the brain, spinal cord, and the retinas. It is my great pleasure to welcome Dr. Huhn with his talk entitled The Clinical Translations of Neural Stem Cells, Transplantations, Emerging Safety and Preliminary Efficacy with Human Central Nervous System Cells, HuCNS-SCs. Dr. Huhn, please.
Thank you very much. Both myself and StemCells Incorporated are very happy to have the opportunity to report what will be a summary of our clinical experience that we've had to date with the human central nervous system stem cell. As you will see, it covers a range of indications that's, I think, reflective of the broad potential of a neural stem cell. And it's even more pronounced or striking because the preclinical data that I'll highlight briefly was all obtained with one cell that's transplanted, not pre-differentiated or modified. I have a limited amount of time, but I'd like to try in that time to give you a very high-level summary of the human data and our translational experience. I'm going to speak in some detail about some of the trials more than others. So very briefly, the human central nervous system cell is an adult multipotent neural stem cell. It's derived by cell surface markers for 133, and it's a rare population of donated fetal brain. It has the self-renewal capacities that we're all familiar with, and it can be cell expanded into master and working cell banks under GMP conditions from which a cell can be derived for human transplantation. And of course, it has both the in vivo and in vitro differentiation into neurons, astrocytes, and oligodendrocytes. So the therapeutic potential of the cell, I think, is rather intuitive. There is a mountain of preclinical data that resides behind this slide, and I don't have the time to go into all of that, but I wanted to highlight the features of the preclinical programs obtained with the cell that have led to the clinical trials that I'll briefly take you through. This work was driven really by a large part of the senior scientists in the company, Dr. Ann Tsukamoto and Dr. Nobuko Uchida. What you see on the upper left is animal evidence of neuroprotection in the hippocampus of the CA1 region in a model of the NCL mouse. Below that, you see the spinal cord injury. This is work done with Dr. Anderson at UCR, where we see evidence of donor-derived interneurons and donor-derived oligodendrocytes in a mouse model of thoracic contusion, which also had behavioral recovery. In a hypomyelination model, the shiver and nod skid, we also see evidence of donor-derived myelination within 6 to 8 weeks. And then finally, on top of all this, the concept of neuroprotection was not lost on us with the opportunity to potentially protect sensory neurons or photoreceptors. And what you see there is the retinal section of our Royal College of Surgeons rat, where you see the engraftment of the human neural stem cell and the preservation of the overlying photoreceptors. So all of this preclinical data, again, there's much more behind this, has led to the successful implementation of initial trials and all of these indications. The first indication, the first in-human study, was a lysosomal storage disease, and there the concept was using the donor cell, the human neural stem cell, as a way to cross-correct an enzyme deficiency by simply transplanting and therefore circumnavigating the blood-brain barrier. It turns out that the two enzymes that are missing in this particular rare disorder are made by the neural stem cell. They're soluble, and they can be taken up by host neurons. Our second indication was Pelletier-Merzbacher disease, which is an X-linked dysmyelination syndrome, and both of these first two brain indications actually are in the setting of a fatal pediatric patient population. In this leukodystrophy, we used the concept of using the neural stem cell as an oligodendrocyte repopulation strategy. We're also interested in CNS trauma, and of course, spinal cord injury has captured all of our imagination, and we have completed studies in thoracic spinal cord injury, which I'll speak to briefly, and now I've initiated studies in cervical spinal cord injury there, and of course, the mechanism, probably putatively, is oligodendrocyte and perhaps even interneuron repopulation. And finally, the concept of neuroprotection, again, I think applies to the eye. The eye has long been held as a particular target for cell therapy. And we've recently completed a study in dry age-related macular degeneration, and I'll announce our phase 1 results later in the talk. This just gives you a scale of what it takes to accomplish these particular studies. We started in 2006 with the first in-human study, which was the first FDA-authorized neural stem cell transplantation. It actually was in a pediatric patient population, but because of the paradigm at that time, it had to be a fatal disease. And not only that, we had to go into the more advanced phenotype of that particular patient population. Again, this was the very first neural stem cell transplantation conducted in the US. We moved on with a second study in PMD, which was completed at UCSF. 6 and 4 patients respectively, because these are ultra-rare diseases. We just need enough patients to understand the safety. We've moved on into the phase 1/2 dry AMD and thoracic studies and completed 15 and 12 patients in those particular trials. Beneath each of those, below the timeline, shows the long-term follow-up. So this is not only a regulatory requirement, but I think a very wise clinical requirement to follow the patients beyond the 1 year to assess safety and other long-term effects that could arise from a stem cell transplant. And again, all these studies were done under either US, FDA, Swissmedic, or Health Canada regulatory authorizations. I want to talk a little bit about our dosing strategy. And as you scale up from an animal model to a human model, you have to make some empirical decisions. Some are based on surgical logistics, some are based on safety, some precedent in the literature. For NCL, because it's a cross-correction, we just had to geographically place the cells as in a wider distribution as we could into the brain. And this just shows the 6 different locations in the cerebral hemispheres, in addition to both lateral ventricles. And the first 3 patients received a total of 500 million cells. The second 3, a total of 1 billion cells into the brain. So we're talking about an effort now to deliver what we think might be a therapeutic dose. In PMD, we were dosing infants because this is a pediatric disorder, so we scaled back our dose a little bit and our number of injections and put 2 sites into each frontal lobe into the deep centrum seminal valley for a total dose of 300 million cells. In spinal cord injury, we're dosing the dorsal half of the cord because that's the most or least eloquent section of the cord to transplant cells, but then rely upon the migratory capacity of the cells to reach the other tracts of the cord. And finally, in AMD, we're dosing just outside the macula into the subretinal space. So that's the nature of how you think about translation and how you begin to think about dosing and the allometric scale up from animal to human. And this is how we ended up for those particular studies. From a safety standpoint, we've now had 2 phase 1s and 2 phase 1/2 studies for a total of 45 subjects. 10 of them were pediatric, 35 were adult. And in our studies, we've had very comprehensive and frequent postoperative clinical assessments by a multidisciplinary team because, after all, this is neural stem cell transplantation. It's invasive. It's in a vulnerable and fragile patient population, so we wanted to have very good assessment of safety in addition to see whether we could see signals of an effect. In fact, at the first study, we had the patients stay at the site for a month after the first transplant for all 6 patients. And we convened a DMC meeting before each subsequent transplant, again, for our very first indication. But now we've collected a broad range of clinical experience, again, in those 45 patients. And most of what we see in terms of adverse events, which is what you report to the regulators, have to do with the underlying disorder or some other endocrine illness. We've had no study-related changes on our imaging that have caused us any concern. The AEs related to surgery and immunosuppression are been the one you'd expect, and they're also mild. We've had the opportunity in the NCL patients to look at postmortem tissue, and we've seen no safety issues with regard to any of the pathological findings or reaction to the cells. And again, we've conducted 4-year studies that are observational after the initial 1 year. Because NCL is a fatal disease, it was not a surprise that we would have the opportunity to look at postmortem tissue, and the families actually were very willing, and in each particular death we've had, they've been more than happy to donate the brain for our examination. So this reflects the commitment of families in a particularly very tragic disease. When we looked at the hemispheres that were frozen by HLA mismatching PCR, we could see evidence of donor cell survival, in this particular case, 2.5 years post-transplant and 1.5 years after stopping the immunosuppression. Because this is an allogeneic cell, all the patients get a temporary course of immunosuppression to, we believe, at least cover the time when the blood-brain barrier has been disrupted, and obviously a bit more there longer. So we found evidence of the cells where we could deposit the cells because we could see the tracks, and we asked the next question, can we see evidence of the cells in the deeper part of the brain? And the deep basal ganglia, which was the slice several centimeters below, we also found evidence of cell survival. We tried to quantify the cell here that you can see in terms of how many we could again, identify in those particular regions. I think the degree of quantification is low, but it's surprising that I have data that I'm not showing now from a patient who's 6 years post-transplant from the NCL study who succumbed to the disease, and the percentage now that we can identify, again, by HLA mismatching is upwards of 3 to 5%. So we're seeing long-term evidence of engraftment survival as well as migration, which I think might be starting to recapitulate the animal findings that we have with neural stem cell transplantation. In terms of PMD, the Plitz-Merzbacher disease, this is a diffuse hypomyelination disorder, and one of the things we were curious about was, would we see evidence by MRI, non-invasively, of de novo myelination against this background of dense hypomyelination? So we looked by DTI, which is one of the MRI metrics to assess evidence of myelination, extremely sensitive, in the regions where we placed the cells, as well as regions remote to that we were confident within a year the cells would not have migrated to. And what we found, and this was published in Science Translational Medicine as the phase 1 results, is that in 3, possibly 4 of the patients, we could see differentiation in terms of the strength of the DTI signal in the regions where we transplanted the cells, and that tended to increase over time in comparison to the remainder of the brain. So this was a sign for us that there was a biomarker that was consistent with our hypothesis of donor-derived oligodendrocytes and myelination. We've moved into thoracic spinal cord injury. This was the first study for spinal cord injury. Typically, you start with thoracic before you move to cervical because of the risk-benefit ratio. This was a phase 1/2 open-label study in 12 subjects. Our eligibility was based upon patients who had both complete and incomplete injury, and I think that's important because I think regenerative approaches probably are going to be more effective if we have patients who have lesser severity of disease. So that was our rationale to include what you see there as Asia B patients. These are patients who still have a motor complete injury but have some sensory function level below the injury. We enrolled patients who were between 3 and 12 months if they were complete, and we thought that window was a little bit longer for the incomplete patients. This was an intermedullary spinal cord injury dose of 20 million cells, above and below the level of injury into the marginal tissue, perhaps intact tissue, that mimicked the data that we had from our preclinical work with Dr. Anderson. As I said, this was an allogeneic transplant, and we transplanted, 12 patients, and each of them got a month of MMF, which is an anti-metabolite, and tacrolimus, and they tolerated the immunosuppression well. The primary objective was safety, of course, but we spent a lot of time trying to understand if we could see signs of a biological change. And in the thoracic cord, because there's no direct motor outflow, we most expected to see this change in terms of sensory gains. So I want to walk you through a summary of how we looked at this. This shows the 7 Asia A patients in the study, the patients with complete injury, and the 5 Asia B patients at the bottom, and each of the sensory modalities that we examined over time at 3, 6, 9, and 12 months for each of those subjects. As you know, these are divided into the different tracks in the cord, and what we found was that a number of the patients had a pattern of increasing gains in the score change from baseline. So this is, again, from baseline to each of those time points. So what we saw was an emerging sign in some of the patients, perhaps more prevalent in the Asia Bs, of sensory gains and across more than one modality. And when you look at the pattern a little bit more closely, we found that 2 of the Asia A patients had an overall pattern of gain. In particular, A2, you notice that there was a deflection in terms of light touch and pinprick, but that's technically because the rating scale says if the patient reports more sensation, you have to count it as altered. But you can see in terms of electrical and heat perception, there was a gain. What's interesting is the Asia B patients, perhaps consistent with the hypothesis that less injured patients would have more of a response, we have a greater pattern of response in terms of their sensory overall gains. In addition, 2 of the Asia A patients converted from a complete injury to an incomplete injury at 10 and 12 months post-trauma. So that was a very significant finding, mostly from a safety perspective to see this kind of data, but also the potential that it represents a signal. So for those modalities, we found that 7 of the 12 patients had changes that we considered significant beyond what you'd expect from their spontaneous recovery. Most patients with spinal cord injury recover most of their function within 3 to 4 months, so you detect things after that tends to look like something that's beyond spontaneous recovery. The changes were gradual, they were not sporadic, and they endured to the end of the 12 months. So again, this speaks more to a biological change. And again, it's consistent with what you might expect from a thoracic injury versus the cervical injury that has more direct motor outflow. It's interesting that the gains were seen not only proximal to the injury, but remote as well, which suggests the possibility of activating long tracts. And again, there was a pattern here. It's too early to conclude that some of the responsiveness appears to be in myelin-dependent pathways of the propriospinal tract versus that of the spinal thalamic. I'm going to end now with our phase 1 data in AMD. This was a multi-center study. Again, the concept here was neuroprotection of photoreceptors, but in a first study, you want to establish safety before you go on to ask more complex questions. The secondary here was assessing, again, just like we did with spinal cord injury, can we, because we're going into the patient population of interest, any signs of change in visual function that looks different than the natural history or any of the objective anatomic measurements of the eye. And the eye in particular offers a lot of objective measurements that you don't have with the brain and spinal cord indications. This is an older patient population, so our course of immunosuppression was shorter, 1 month of MMF and 3 months of tacrolimus. It was dose escalation, and because this was not perhaps first in human, but first in location with the FDA, they wanted us to go into patients who had very severe vision in cohort 1, where we did our dose escalation. So that's 2,400 or worse, so that's beyond legally blind, who had areas of GA, which refers to geographic atrophy, or the amount of RPE defect, which I'll explain in a minute, as being quite large in many of those patients. We tested 200,000 cells in the first 4 patients, then went to 1 million cells thereafter, and then in the second 7 patients in the study, or the cohort 2, we went now to patients who had a little bit better vision. What we found, not surprisingly, is that one of the metrics of visual function is best corrected visual acuity. That's the eye chart. This shows the study eye outcome over 12 months and the fellow eye. And the nice thing about the eyes is you have the ability to dose one eye and look at comparisons in the contralateral eye. And in dry AMD, much of the disease pathology is congruent between both eyes. The study eye had to be the worst vision of the two by protocol. And this just shows you generally that we had an overall improvement or stability in both eyes. And that's not unexpected in a disease that has a slow progression of visual loss. In addition, patients with that poor vision have a high learning capability on some of the tests, and so ability to interpret this is confounded by that particular attribute. But from a safety perspective, it's very reassuring to see the stability, if not signs of improvement, in the majority of patients. BCVA is not the only metric by which you can assess vision. There's a lot of other visual qualities that we don't think about, but we rely upon, and one is contrast sensitivity. This is what helps us drive at night. It helps us go down a dimly lit stairwell. And we found that a majority of the patients actually had a gain in their contrast sensitivity. And you can see, this is the chart on the left, how you acquire that in the clinic, is that you determine which letter they can see and where it stops. And that's a measure of their visual contrast sensitivity. Some of the patients, I think, had their natural progression, but in none of them did we see any precipitous change if they had worsening. So this is a very interesting finding in terms of a very first open-label study to see a gain of one of the important visual metrics. The underlying disease in AMD is geographic atrophy. That's loss of the RPE, and this just shows you the distinction and what an inhomogeneous patient population can do to a phase 1 trial. On the right, you see a patient who has fairly uniform, very simple area of RPE defect, which is shown by fundus autofluorescence. And then in pink is the computer's attempt, semi-automated way to calculate what that area is, because the area is one of the metrics by which you assess disease progression. Now, our animal data showed that our effect was on the retina, not the RPE, but we were curious to make some observations in this study about this particular outcome. Compare that with another patient in the trial on the right, who has much larger, much more complex GA as a result of the requirement of needing patients in the study who had very poor vision in our first cohort of patients. What we found by measuring GA was that there was a change in our prospective analysis that was conducted during the study that suggested or indicated that the rate of progression in the study eye was much better than that of the fellow eye. But as you can see, this is complex data to analyze and to assess. We were enough interested in this particular observation that we conducted a second analysis. The post-hoc analysis, which is part of clinical development, did not reveal a similar trend, but right now there's an inconsistency between our readings of this one particular endpoint that we're not going to make any conclusions until I get a chance to spend a little bit more time with the data, in part because this data all came out just about 5 days ago. But I want to disclose that this is part of the complications of when you do clinical studies, is beginning to understand the signals and interpreting the data. What is interesting about this is, though, in terms of another anatomic metric that you can measure, and again, this speaks to the appeal of studying the eye, particularly for cellular therapy, is you have these objective measures. This is a measure by which you can determine the thickness of the fovea and the volume of the macula by ocular coherence tomography. This is an objective measure, and this plots the study eye versus the fellow eye for the central subfield thickness, which is the foveal thickness, and that of the macular volume, again, in the study eye and the fellow eye. And for each of the 3 cohorts in the study, the 200,000 cells, the first 1 million, and then the 7 patients who had the additional 1 million, that there's been an increase in their thickness in the study eye and an increase in their volume for the macular structure versus the fellow eye, which has actually gone down, which is more consistent with the natural history. So this is a very intriguing and perhaps important finding in the phase 1/2. As all part of studies, there is a risk-benefit, and I didn't go into details of the adverse events for the other studies, but because this is our first disclosure of the AMD data, I'll take you through what we've been able to see. Most of the adverse events have been related to their underlying or any comorbid conditions they had. In terms of surgery, we expected to see cataract formation and some mild transient retinal detachment. None of this was actually of concern to the patients or the investigators, and it's expected outcome of a post-vitrectomy state, and you have to do a vitrectomy to get access to the subretinal space. It's interesting and perhaps relevant that our immunosuppression course, we expected that we'd have issues with the geriatric population, and we did in terms of their electrolyte disturbances, but this could be managed through the immunosuppression experts. And again, in the age population, the renal sensitivity is higher to this, but it's something to think about as we pursue allogeneic approaches. In terms of this cell, we had no serious ocular events related to the cell itself, which is always comforting as you think about developing a program, and in fact, the cell was very well tolerated. So we came out of the AMD study with a sense of safety and tolerability and feasibility, that the BCVA was stable or improved, that the contrast sensitivity was, I think, very intriguing to us, as was the changes by OCT. And all of this is the first step that you do as you begin to develop something and move into the phase 2. So I'll finish right now. I know I've rushed through all of this quickly. It's 10 and 15 years of preclinical and clinical data to try to go through quickly. And there's probably no doubt many ways to implement a clinical and developmental program, but I think the first 2 steps are showing and launching everything based upon very strong in vivo data, which unfortunately I wasn't able to take you through, and then develop a good database of safety, tolerability, and feasibility, because these are invasive applications, but they're all for diseases that have tremendous unmet need and no current therapy. The pattern that we see when we look across all of our studies is that of emerging biological activity and preliminary efficacy, and I think we can all agree that the first clinical signs of an effect are likely to come incrementally and slowly, and I think you've been able to see that here in my presentation today. We've seen evidence of cell engraftment by postmortem tissue, which I think is a very important milestone. We see evidence of biomarker changes consistent with our hypothesis in the PMD study. And finally, we're beginning to detect signs of clinical changes that could have particular relevance as we go from the thoracic to the cervical region. All of this allows us to move the field, at least with regard to the human central nervous stem cell, into controlled randomized studies. There's a phase 2 now underway for cervical spinal cord injury. That's been labeled the pathway study, and another one now that's actually initiated for dry AMD, which we will compare outcomes from the control eye to the study eye as the RADIANT study. So again, thank you very much. There's, as I said, a mountain of data behind all of this, and these are our selected publications. Thank you.